Alright, hi year 12s, Mr. Lim here again, and this is the second video on tertiary structures of proteins. Okay, so tertiary structures of proteins. Remember that we are combining side groups, or talking about interactions between the side groups of amino acids, not the peptide bonds of the amino acids, which differentiates it from secondary structures. Okay, so dipole-dipole interactions are formed when two side chains of amino acids, which are polar but not polar enough for hydrogen bonding, create a force holding them together. Okay, so serine and methionine is an example of this. Methionine, the side group, I'm not going to draw the rest of it, but the side group is made up of CH2, CH2, S, CH3. So this would have generally a negative end, that's sulfur. Okay, and then a side group of the methionine, no, we did that, the serine would be a C with a CH2, oops, CH2, and then an OH. Okay, and so that negative end is that positive end, so this positive end here, right, the positive end on the hydrogen and the negative end on that oxygen, but the positive end of that hydrogen would form a dipole-dipole bond with the negative end of that sulfur, and then hold that together. It's not generally a very strong one, but it does exist, so therefore we have to learn about it. Okay, these interactions are quite weak, but a large number of them can do their thing. All right, dispersion forces. So, also known as hydrophobic forces. So, the idea is that side groups are either polar or non-polar or a mix of the two. Okay, and so here are some non-polar side groups. Okay, so you have the amino acid end part of it, and then the part in blue is the non-polar parts. Okay, so all of them are just made out of carbons and hydrogens. They're all generally non-polar. So the idea is that some proteins need to be soluble in non-polar substances, and some need to be soluble in polar substances. And so if you need to have a protein that is in a non-polar solvent, so like in fats, the non-polar side groups will be found on the exterior of the protein. So remember, here we have our protein and it's all folded into a particular shape. But your non-polar, which will be in blue, side groups might be here. All the non-polar side groups. Okay, all the non-polar side groups will be on the outside of your protein. And this is a spherical thing, but all the non-polar things will be on the outside. And all the polar side groups will be on the inside. Okay, so all your polar side groups will be on the inside, sticking together in the polar end. All right, so that's if you want it to be in a non-polar solvent like a lipid bilayer. All right, but however, if you need to be in a polar solvent like water, it'll be the opposite. So here's your folded protein. Okay, and then all of the polar side groups will be on the outside. All of the polar side groups will be on the outside, interacting with the water. Polar side groups on the outside, and all of the non-polar side groups will be on the inside, trying to stay away from the water. Okay, so all the non-polar side groups on the inside. Okay, and so by having this, you maximize the interactions which are best suited for that. Okay, so the hydrophobic and hydrophilic nature of the side groups can provide a significant amount of force to keep the protein in correct shape. So generally, the non-polar stuff generally sits on the inside for polar soluble proteins, and they all kind of sit in the inside and they hold together because they don't want to be accessed by the water on the outside. All right, and then finally the tertiary structures. Right. So the tertiary structure, we have ionic interactions as the last one. So certain amino acids that have side groups that can be changed into ions due to functional groups on the side groups. So in other words, anything with an amine or a carboxylic acid can be turned into a ion. All right. So aspartic acid is an example with lysine. Okay, so there's the aspartic acid side group or side chain, right? Oh, actually, that's the lysine one, sorry. And then the aspartic acid one is here. Okay, and so this one forms the COO minus, that forms NH3 plus, and so therefore they're going to have an ionic interaction between there, the positive and the negative thing there, electrostatic attraction there. Okay, so that's going to perform there. Alternatively, you could even have a sodium ion interacting with the aspartic acid. Okay, so the aspartic acid is a CH2COO. O minus, okay? And so there'll be an interaction between the sodium ion and the carboxylate part of that side group, okay, assuming that it's lost its hydrogen there. These ions can interact with other ions or other ionic side chains to perform various roles, such as one ion can have ionic interactions with a number of amino acids in the same protein, or over multiple proteins to ensure that they stay together. So what that means is that instead of this being a negative and a positive, what it might be is that, okay, here's my iron ion, like in hemoglobin, okay, and then you have a whole bunch of, this is one carboxylate ion from one part of the chain, and here's it from another part of the chain, another carboxylate ion from one of the side groups, and then effectively that has an ionic interaction, that has an ionic interaction, and therefore these two side chains are held together. Okay, and so that one ion can hold together things that are quite far apart, and that's what happens with most of the ions there. The hemoglobin is one iron ion holding together like 4 heme units, all into one area to hold it all together and doing its thing. All right, or you can just have a positive end and a negative end holding everything together for that part. Okay, but this one's quite more fun. But anyway, that's the idea. The ionic charged parts hold stuff together, and therefore they can't break apart, and everything gets held together, holding it into shape. That's it.